Hello friends, myself Professor B.D. Bhoknal, Associate Professor, Botany Department, KTGM College, Nasik. Today's topic is from Cell Biology and Molecular Biology, paper 2nd. From that, cell organelles. Now today's topic is structure of chromosome. Within that, morphology of chromosome. Morphology of chromosome is best studied during metaphase and anaphase stages of mitosis. The size of chromosome varies from one organism to another. Further, the chromosome in a cell are never alike in size. Some may be exceptionally large while others may be too small. The largest chromosomes are lamb-brush chromosomes present in certain vertebrate oocytes. However, most metaphase chromosomes fall within a range of now 3 millimicron in Drosophila to 5 millimicron in man and 8 millimicron in maize. The shape of chromosome is determined by the position of centromere. Chromosomes are classified into 4 types according to their now shape. First, telocentric. The rod-like chromosome having the centromere located on the one end. Second, acrocentric. These are also rod-like chromosome that have very small or even now imperceptible short arm. Third, submetacentric. The submetacentric chromosomes are J or L shaped, having two arms of unequal length. The centromere is located away from center. Fourth, metacentric. The V-shaped chromosome having two equal or almost equal arms. The centromere occurs at the center. Now see within these types of chromosome. Now first, telocentric. Now in case of telocentric, it has one long arm and the centromere it is present towards the one end. It look like now eye shape. In acrocentric type of chromosome, it generally shows two unequal arm. The one arm which is longer in size and another arm which is now short arm. Then in case of submetacentric, chromosome, it generally look like now L-shaped. Now one arm which is short and another arm which is now long. Then metacentric. Now in case of metacentric chromosome, it look like now V-shaped. The two arms are somewhat equal in size. Next, structure of chromosome. The chromosome has generally 8 parts: centromere or primary constriction or kinetochore, chromatids, chromatin, secondary constriction, telomere, chromomere, chromonemata, and matrix. Centromere. It is the primary constriction at the center to which the chromatids or spindle fibers are attached. Its function is to enable movement of the chromosome during the anaphase stage of cell division. Chromatids. During the cell division, A chromosome is divided into two identical half strands joined by a centromere. A chromatid is each half of the chromosome joined. Each chromatid contains DNA and separate at anaphase to form a separate chromosome. Both chromatids are attached to each other by the centromere. Chromatin. It is a complex of DNA and proteins that forms the chromosomes within the nucleus of eukaryotic cells. Nuclear DNA is highly condensed and it is wrapped around nuclear proteins in order to fit inside the nucleus. In other words, it is not present as free linear strands. The chromatin consists of DNA, RNA, and now the proteins. Secondary constriction. It is generally present for the nuclear organization. Telomere. Telomere is the terminal region of each side of the chromosome. Chromonema. It is thread-like coiled filamentous structure along which chromomeres are arranged. Chromonema control the size of chromosome and it act as a site of gene bearing. Chromomeres. These are the bead-like structure present on threads or chromonema. These are arranged in a row along the length of chromonema. The number of chromosomes is constant and it is responsible for carrying the genes during the cell division to the next generation. Matrix. Pellicle is the membrane surrounding each of the chromosome. Matrix is a jelly-like substance present inside pellicle. It is formed of non-genetic now materials. Now see within the structure of chromosome diagram. Now at the top, now the pellicle, then the secondary constrictions, matrix, then now the spindle fiber at the base, at the middle primary constriction, then now chromonemata, and at the end now is satellite. These things are present within the structures of chromosomes. Next point, number of chromosomes. The number of chromosomes in plants or animal species is fixed. Generally, the somatic cells of animals and higher plants are diploid, while gametes have haploid chromosome number. Gametes normally contain single set of chromosomes. This number is called haploid number. Somatic cell usually contain two sets of chromosomes. Hence, it is called as diploid. The haploid sets of chromosomes known as genome. The lowest chromosome number recorded in eukaryotes is 2 in now the mesotoma, that is now flatworm, and the highest chromosome number recorded in now these Alacantha, that is now from protozoa, in which diploid number is approximately now 1600. Again in the plant kingdom, the plant Ophioglossum, now reticulatum, that is from fern, have the highest chromosome number, that is now diploid is equal to 1260. Among angiosperm, only 6 species are known to possess the lowest identified chromosome now number, that is diploid chromosome number is equal to now 4. These plants are the monocotyledons, now the zingeria, then now the colpodium, then now Rhynchospora, etc. from now the dicotyledons. Regarding the number of chromosome, take example of now the human being. It consists of 46 diploid chromosome and 23 haploid chromosome. Then take example of frog. It consists of now 26 diploid chromosome and 13 haploid chromosome. Then take example of mosquito. It consists of 6 diploid chromosome and 3 haploid chromosome. An onion consists of 16 diploid chromosome and 8 haploid chromosome. Next example is tomato. It consists of 24 diploid chromosome and now the 12 haploid chromosome. Another example is now the pea. It consists of now the 14 diploid chromosome and 7 haploid chromosome. Then cat. It consists of 38 diploid chromosome and 19 haploid chromosome. Then in case of now the dog consists of 78 diploid chromosome and 39 haploid chromosome. Then the another example from sunflower. It consists of 34 diploid chromosome and 70 now haploid chromosome number. Next point now, that is ideogram. Ideogram is the diagrammatic representation of karyotype showing all the morphological features of the chromosomes grouped on the basis of position of centromere and order in series of decreasing size. Chromosome banding and now the painting technique are used to identify each chromosome. These techniques are also used to distinguish each arm of the chromosome and to investigate specific region within them. In some chromosome, the centromere, which divide each chromosome into long and short arm. The long arm is denoted by the letter P and the short arm is denoted by letter now Q. For example, the short arm of chromosome 5 is denoted simply writing 5P, and thus in the short arm of chromosome 5 we have region 5P11, which is closest to the centromere, followed by region 5P12, 5P13, 5P14 and 5P15, which is the farthest from the centromere. Within each region, individual bands are denoted by numbers following a decimal point. For example, 13.1, 13.2 and 13.3 refer to the three bands that make up region 5P13. The pattern of bands within the chromosome is called an ideogram. Next point, that is karyotype. Karyotype. The word karyotype is derived from Greek word now, that is kernel, which means the contents of nucleus. The term now karyotype is referred to a group of characteristics that allow the identification of particular chromosome pair on the basis of general morphology of a set of chromosomes at metaphase of somatic cell of an individual. Karyotype concept was derived by now S. Nawashin based on the observation that the number of chromosomes and morphology of each chromosome pair is normally constant and characteristic for a species. Karyotype concept is phenotypic appearance of a chromosome and is homologous pair which helps to differentiate types of chromosome based on their morphology in an individual species. Karyotyping is a method of arrangement of pairs of homologous chromosomes of a cell in decreasing series of their size. Karyotype study is very important to identify abnormalities in chromosomal structure and now the number. Karyotype study also helps to determine the origin and the evolutionary relationship among the different taxa. Thank you, and that's all.